Alright guys, welcome to OTC episode number 3. In this episode, we are going to discuss about the details of development of different nucleus of the lens. We are going to discuss about the blood supply of the retina, the blood retinal barrier, the blood supply of optic nerve, and a little bit of the ocular structures development. Yes, okay, so now let's start. So what happens is, we have done that the lens develops from surface ectoderm. Remember, so there was optic stalk, optic vesicle, optic cup. There was surface ectoderm which was anterior to the optic cup, which got invaginated and separated the single cellular layer that form the circular structures. And now what happens is, so that circular structure which is formed, it lies between the surface ectoderm and the optic cup. Now this circular structure which is here, I'm just going to show you a quick image. So this is a single cellular structure. Now just give me a second, I'll show you something so that it becomes easy for you to connect the dots. Okay, so check this out. Now can you appreciate that there is lens vesicle, and you can also see that in due course there is movement of the posterior cells to the anteriorly. Now initially the cells are cuboidal in nature. You can see here, yes. So when they move from posterior to anterior, so they become what? They become columnar. So when they become columnar, because they are getting stretched, right? The nucleus also moves from posterior side to anterior side, such that on the anterior side they form a bow-like structure. Bow, you understand? Bow and arrow, tir kaman, that command thing. So they form a nuclear bow. So you can appreciate that there is anterior nucleus arrangement, then there is another arrangement of the nucleus. But my dear friends, this nucleus has been derived from the stretching of the cells which are cuboidal initially. Now they have moved anteriorly, forming columnar configuration, and the proteins which were initially in these cells, they turn into transparent proteins. Those proteins are known as crystallines. Okay, now I think we can write here. So you can appreciate which proteins are these. These are your crystalline proteins. Crystalline proteins, and they are transparent proteins. If we are talking of lens, it has to be transparent. Now once there is movement from posterior to anterior, so this is going to form one nucleus of the lens. So lens is divided into three things. One is the central nucleus, the peripheral cortex, and the capsule which surrounds the lens. Okay, so if we talk about the nucleus of the lens, so here I am going to write nucleus. So from the core to the periphery, so first we have got embryonic nucleus which is formed initially. Then second, we have got fetal nucleus. So what happens is, once there is movement of the cells which are present at the equator, so they remain active throughout their life. So this is the equator of the lens. This is the anterior surface, this is the posterior, this is the equator. So the cells which are there at the equator, now they start forming fibers anteriorly as well as posteriorly, anteriorly as well as posteriorly, anteriorly as well as posteriorly, such that the central nucleus, embryonic nucleus, which was initially formed, it gets detached, yes, from its surrounding region, and there is concentric formation of the more and more fibers. Okay, now you can see. So if this is your embryonic nucleus, so the peripheral equatorial cells, so they start forming the fibers anteriorly as well as posteriorly. So then after embryonic nucleus in the core, there is fetal nucleus, and outside of the fetal nucleus there is infantile nucleus, and outside the infantile nucleus there is adult nucleus. Now when there is development of the fibers in the fetal stage, so it goes in a way such that there is formation of anterior Y suture, and there is also formation of posterior Y suture. So you can appreciate that the sutures are formed when the fibers are spreading in the fetal stage. In the fetal stage, so the sutures will merge such that they are going to form erect Y anteriorly and inverted Y posteriorly. Fetal stage, when the fibers go. Alright, so this is the formation of sutures that you have while development of the lens. So if I draw the lens, so central part is my nucleus, while the periphery is the cortex. So you know that the most central part is formed first. So embryonic nucleus is formed during 0 to 3 months of gestation. Fetal is 3 to 8 months of gestation. Infantile is 8 months to puberty. And adult is puberty to adult stage. And surrounding this there is cortex, and once the cortex is also formed, so there is secretion of strongest basement membrane in the body, and that is your capsule. So lens is enclosed by a capsule, which is the strongest basement membrane of the body. ठीक है जी? Okay. Now I am again drawing the structure of cornea. This is your lens which has got zonules, and this is your posterior segment. We had done it in our second episode that anterior segment is from posterior surface of the cornea till the anterior surface of lens, while posterior segment is from posterior surface of lens to the anterior surface of retina. Okay, now we have already done about the hyaloid artery which is present, which supplies the lens. Okay, so there is presence of hyaloid artery. I am just going to tell you. So during the development of the lens, so there is hyaloid artery. So if this is your optic cup, I'm just going to repeat it. So this is your lens which is developing. This is your surface ectoderm. Now lens is vascular or avascular. Since lens is avascular, so it has to derive its nutrition. So from where it is going to derive its nutrition? From one artery which is your hyaloid artery. Which artery? Hyaloid artery. Now this hyaloid artery surrounds the lens completely. Yes. Now I am going to show you another beautiful image to connect the dots. Just give me a second. Okay. Okay, so this is, you can appreciate that there is the central nucleus and peripheral cortex, and there is anterior capsule and posterior capsule, and the peripheral part of the lens is equator. Now coming over to hyaloid artery. So if you could appreciate, you just need to listen, then we'll later write. So there is hyaloid artery which is arising from the optic cup. So it is surrounding the developing lens. Initially the lens is sphere in shape and later it turns from sphere to biconvex. Now this hyaloid artery further forms three branches. As you can see in the image, there is posterior pupillary membrane. Oops, the ball fell. Posterior pupillary membrane. Second is capsulopupillary portion, and third is anterior pupillary membrane. So it is completely surrounding the lens so that the lens is able to attain the nutrition during its development. But this hyaloid artery later regresses. In some patients, it fails to regress. Then there is a problem. What problem is there? I am going to show you the pic, then we will write. If there is complete remnants of the hyaloid artery arising from the optic cup reaching up to the lens, you call it as PHPV, persistent hyperplastic primary vitreous. If the hyaloid artery gets regressed but in parts, you call it as musci volitentis. Why musci volitentis? Musca word is used for housefly, Musca domestica. Volitentis means flying. So because of the incomplete resorption of hyaloid artery, the parts of the hyaloid artery which are left, they are going to cast a shadow on the retina. Now this shadow will be appreciated by the patient as if there is some fly which is flying in the visual field of the patient, when actually it is not in the external visual field, it is the shadow of the incompletely resorbed hyaloid artery. Third is, if the origin part, small stump of the hyaloid artery is left at the just origin site, you call it as Bergmeister papilla. You can see here, check out the pic, Bergmeister papilla. If the small part which is getting inserted posterior to the lens is left, rest entire hyaloid artery is resorbed. That is your Mittendorf dot. I think you can appreciate Mittendorf dot. Okay, so these are the features that you see. Now once we have understood it, now let's revise it while writing. So hyaloid artery has got three parts. So one is posterior pupillary membrane, as we see in the image. Then there was capsulopupillary portion, then anterior pupillary membrane. So it was supplying the blood to the developing lens, and lens is avascular. Now incomplete resorption, incomplete resorption will lead to four things. One is PHPV, that is a short form which is used, which is persistent means it is persisting, persistent hyperplastic primary, P for primary vitreous. Yes. Second is musci volitentis, musci volitentis. So this flying thing that the patient sees, that is known as floaters. I hope it is clear. Third is Bergmeister papilla, Bergmeister papilla. So since I told you there is Bergmeister papilla, which is just the stump which is left at the origin where it lies at the optic cup. Now because it is lying at the optic cup, any stretch to the Burgmeister papilla will lead to retinal detachment, because it is attached to the retina na. Retinal detachment, that is one important point. Okay, fourth is the one point which lies in close contact with the posterior surface of the lens, that is Mittendorf dot. Mittendorf dot. Because it is attached to the lens, it will lead to cataract. But now since you have understood this, PHPV is complete persistent. That means it is at the rising point and it is also at the inserting point, so it can lead to both retinal detachment and cataract. RD is the word which is used for retinal detachment, and usually this is unilateral condition. Usually, just one extra point, if PHPV is there, it is usually unilateral condition. So this is about hyaloid artery. Okay, now getting back to here. Okay, now we have done about around the ciliary body, what happens, around the iris, what happens, and there is your cornea. Now coming over to how the cornea is formed, please focus. So we have read about the layers of the cornea. Now it will be very easy for you to connect the dots. So there is optic cup, है ना, and anterior to optic cup there is surface ectoderm. Now this optic cup is closing around the lens, so that it will come to lie like this, and ultimately this blue part which I am forming is your iris. Okay, now we also know that the entire optic cup is bathed in mesenchymal tissue, so it is all mesenchyme, है ना. Now what happens is there is first wave of mesenchyme that comes to lie between the surface ectoderm and developing iris. This is your first wave. Then there is second wave which comes to lie between surface ectoderm and first wave of mesenchyme. Okay, all right, I am going to show you the image one more time so that you are able to further appreciate the structure that I am willing to show you. Check this out. So you can see that there is one wave which is marked as 2, that is your second wave, and that is your first wave, and one wave which is marked as 3. Yes, okay, so the one wave which is marked as 1, that is your first wave, and one wave which is marked as 3, that is your second wave, okay. Now these two waves, they are going to form what? They are going to form the inner layers of the cornea. So surface ectoderm forms the epithelium of the cornea. What are the other layers from anterior to posterior? So from anterior to posterior we have got anterior epithelium, then we have got Bowman's layer, then corneal stroma, Descemet's layer, and endothelium. Descemet's layer, so there is one Descemet's layer. There is also newly discovered layer which lies anterior to the Descemet's layer, that is Dua's layer, pre-Descemet membrane. So the first wave forms the posterior two layers, that is your Descemet's membrane and endothelium, while the second wave forms the Bowman's layer, stroma, and pre-Descemet's layer, because some believe that pre-Descemet's layer or Dua's layer is nothing but the modification of posterior most part of the stroma only. Now this is formed from mesenchyme, and I have told you that mesenchyme may take form of mesoderm or even neural crest. Here it is actually the neural crest. Okay, so anterior surface of the cornea is formed by surface ectoderm, while the rest of the cornea is formed by mesenchyme, which gets transformed into neural crest cells. I hope it is clear. Now I am upping the game a little bit more. Now check this out. So once the cornea is formed, so now coming over to the formation of eyelids and lacrimal glands and conjunctiva. So what happens is, the most peripheral part of surface ectoderm, it evaginates anteriorly and it meets somewhere at the middle. Yes, and later it separates. So you can appreciate that there are ectodermal folds which is coming from the peripheral part of the surface ectoderm, which later forms eyelids. So if I may ask you, eyelids are formed by, it is surface ectoderm. If I may ask you, lacrimal gland is formed by, because lacrimal gland, so this is the normal cornea which is formed, surface ectoderm, and this is the ectodermal fold which is coming forward. So there is a junction where there is lacrimal gland. So it is actually the junction where it is formed, as you can see the buds of lacrimal gland. So lacrimal gland is also formed at the surface ectoderm. And because the inner part of the eyelid consists of 7 layers, the innermost part of the eyelid is covered by conjunctiva, which is actually the 7th layer of the eyelid. The conjunctiva is also formed by surface ectoderm. So let's revise. So cornea, anterior surface, surface ectoderm, rest of the layers mesoderm, which gets transformed into neural crest. The same mesoderm which was forming the first wave, descemetes and epithelium, the second wave, Bowman's, stroma and Dua's layer, pre-descemetes layer. Then there is ectodermal fold formation, ectodermal fold which form eyelid, which is again surface ectoderm. Then there is lacrimal buds, conjunctiva, which is again surface ectoderm. Clear? Okay, now once these structures are formed, you can appreciate. So hyaloid artery is primary hyperplastic vitreous, which is left if it is remained, if it fails to resorb. Now once the cup is closed with lens posteriorly, so the hyaloid artery has to resorb. But when it fails to resorb, PHPV. But when it resorbs, so after that there is a remnant of a canal in which the hyaloid artery was passing. That canal is, as you can see in the B image, it is your Cloquet canal. And around this canal there is what? There is which structure? There is optic cup. The optic cup secretes a fluid which later comes to be vitreous humor. So if I may ask you, what is the embryological derivative which forms primary vitreous humor? So primary vitreous was nothing but remnant hyaloid artery. So that is your mesoderm. But if we talk about secondary vitreous, so it is not mesodermal in origin. It is secreted by neuroectoderm. Why? Because optic cup is neuroectodermal in origin. Is it understandable? That is your secondary vitreous. And the lens zonules, which is thread-like structure that holds the lens at its place, that is tertiary vitreous. That is also secreted by optic cup and hence neuroectoderm. So secondary vitreous, tertiary vitreous, as you can see, it is neuroectodermal in origin. But primary vitreous is actually hyaloid artery. It is mesodermal in origin. We are going to write it now. Okay, let's write. Please write. So primary vitreous humor is mesodermal in origin, while secondary vitreous humor and tertiary vitreous humor is neuroectodermal because they are being secreted by the optic cup, which is neuroectoderm. Optic cup. All right. So we have done the embryology of eye completely. Any doubts, you can get back to me anytime on social media. I have also shared my WhatsApp contacts. Okay. Now coming over to the retina. For retina, we know that the optic cup forms neurosensory layer. The inside and outer side is retinal pigment epithelium. So later there is fusion and it is forming the 10 layers of the retina in due course. Now let's do histology of retina. So there are 10 layers of the retina. The innermost layer is not shown here, that is inner limiting membrane. I had told you in episode 1 while we were doing development of ciliary body, in which there is internal limiting membrane which forms the innermost layer of the ciliary body. So inner limiting membrane or internal limiting membrane is your first layer. Then there is nerve fiber layer. So you can appreciate that there is nerve fiber layer. Nerve fiber layer is the same layer that actually forms the optic nerve, which exits the optic disc region, the neuroretinal rim. And third layer is ganglion layer, ganglion cells. Now ganglion cell nuclei can be easily seen in this image. Now if there is one cell who has got nucleus and there is another cell which has got nucleus, how the cells are going to get connected? By the means of synapses. So there is ganglion cell whose nucleus is visible to you, and there is another layer which is your inner nuclear layer. Why it is known as inner nuclear layer? Because it comprises of nuclei or nucleus. Now how to connect the ganglion cell layer with that of inner nuclear layer? By the help of synapse. You call it as plexus, hence the name inner plexiform layer, because it lies inside. Now there is inner nuclear layer, so there is outer nuclear layer as well. How to connect inner nuclear layer and outer nuclear layer? By the means of plexus. So there is outer plexiform layer and there is outer nuclear layer. So easy. So far we have been able to do 7 layers of the retina. Now if there is internal limiting membrane, of course there has to be some external limiting membrane. 8 layers, and one layer is what we have been reading since our childhood, that is rods and cones. So that is our 9th layer. And during the development of the optic cup, the inner layers were forming neurosensory layer, while outer layer was forming the lone layer, that was retinal pigment epithelium. So it is complete 10 layers which we have just done. And outside of the retina there lies one membrane that is known as Bruch membrane, and outside Bruch membrane there lies choroid. Then we have got sclera. I have already told you about the layers of the sclera. I have already told you what lies outside the sclera. This is Tenon's capsule. Outside Tenon's capsule there is conjunctiva. Okay, all right. So this is about the histology of retina. Now talking of the blood supply of the retina. So far we have done the blood supply of developing lens. Yes, so blood supply of the eyeball we have done in first episode. Okay, now what happens is the inner 2/3 of the retina, inner 2/3 means till outer plexiform layer, the inner 2/3 of the retina receives its blood supply from central retinal artery, CRA, central retinal artery. I'll write it here for you guys. Central retinal artery. But the outer 1/3 of the retina, it is outer 1/3, it receives its blood supply directly from choroidal circulation, the same circulation which was being formed by short posterior ciliary arteries. The 10 to 20 short posterior ciliary arteries which were piercing the sclera were entering into the choroid and forming choriocapillaries. If you could recall, we had done it in the blood supply of uvea, choroidal circulation. But did you notice one thing? This outer plexiform layer receives the blood supply partly from the central retinal artery and partly from the choroidal circulation. This layer is known as watershed layer, watershed layer, or Henle's layer, Henle's layer. So it is having dual blood supply. I am writing it here, dual blood supply, and here lies choriocapillaries, from where there is exchange of metabolic waste products from the outer 1/3 of the retina. This is about blood supply. But if we are talking of blood supply, we have to know about BRB. BRB, not be right back, not slang. It is blood retinal barrier. So there are two blood retinal barriers. Two blood retinal barriers. One is your inner blood retinal barrier, inner. Another is your outer blood retinal barrier. So inner blood retinal barrier is formed by endothelium cells, endothelium, which is surrounding the, which is the forming the capillaries, and there are pericytes which is surrounding the endothelium cells. Endothelium cells and pericytes, they are present where? Around the capillaries, which are nothing but central retinal arteries forming this, है ना? Outer blood retinal barrier is retinal pigment epithelium. So there are two blood retinal barriers. So anything which is entering into the retina cannot enter without the permission of inner blood retinal barrier if it has to enter via central retinal artery, or outer blood retinal barrier, which is retinal pigment epithelium, if it has to enter through choroidal circulation. So this is about blood supply of the retina. Now next is your blood supply of optic nerve. We'll just do a little bit of touch and go. Now we divide the optic nerve head. So I am talking of the blood supply of optic nerve head. So optic nerve head is divided into three main parts. One is prelaminar region, one is nerve fiber region, and third is retrolaminar region. So I am going to tell you. So nerve fiber region and prelaminar part is this. So this is your this is your prelaminar part and superficial nerve fiber region. Then second is lamina cribrosa. What is lamina cribrosa? Lamina cribrosa is the place, is the part of the eye where the optic nerve pierces to enter into the eye. Then third is retrolaminar region. Now let's understand this. So since I told you the superficial, so superficial is supplied by central retinal artery. I have already written, I am going to highlight this. So superficial is your parts of central retinal artery, cilioretinal artery, and then there is peripapillary choroidal vessels. Can you appreciate? Peripapillary choroidal vessels, and this is your central retinal artery. Okay, now cilioretinal artery is actually the branch of short posterior ciliary artery only, SPCA. If you could recall, 10 to 20 short posterior ciliary artery vessels, they were piercing the sclera. So that is cilioretinal artery, and also it will be supplied by central retinal artery, and there are vessels of Zinn and Heller, which forms the entire circle around the optic nerve. Yes. So this is about the nerve supply of nerve fiber layer or superficial and also prelaminar region of the optic nerve head. Done. Now second, which is lamina cribrosa. So lamina cribrosa, and you can appreciate this is your short posterior ciliary artery. I'm just highlighting it for you. There is lamina cribrosa, which is again supplied by, which I have just highlighted, short posterior ciliary artery. And lastly, we have got the retrolaminar region, which is supplied by pial vessels as well as central retinal artery. Can you appreciate the pial vasculature as well as the branches of central retinal artery? Okay, so you can easily appreciate. So this is your central retinal artery, and you know central retinal artery is a branch of ophthalmic artery. So this is about the blood supply. I am just projecting it here so that you get it. Blood supply of optic nerve head. So we are mostly concerned about the blood supply of optic nerve head. The optic nerve head is the part which is lying inside the eyeball. You also call it as intraocular portion of the optic nerve. And how thick it is 1 millimeter thick. So if we talk about the optic nerve head, so there are 4 parts of optic nerve head. One is intraocular portion, second is retrobulbar part. Retrobulbar part is the one which lies behind the eyeball, and third is the part which lies in the optic canal, so you call it as canalicular part, and fourth is the part which lies inside the cranium, you call it as intracranial part. So the length of the optic nerve in the intraocular portion, or in other words the optic nerve head, is 1 millimeter. The retrobulbar part is nearly 15 to 25 millimeter. It varies from person to person. The optic canal part is nearly 7 to 8 millimeter, while intracranial part is 8 to or 9 to 11 millimeter. So the usual length of optic nerve is 35 to 50 millimeter. But one thing remains constant, that is the intraocular portion of optic nerve, which is optic nerve head.